Now let's practice what you have learned. Here you have several points to remember. The practice requires you to pay attention to details that you went over in the previous parts. First, the live console allows you to try running code. The first assignment is to simply check the code syntax. Notice that if you just run what's already written, it will not work because the syntax is broken. The challenge is to fix the code by adding small changes that will make it work. Refer to the previous section to make sure that you understand how the code should be written. You will get a message that the code worked correctly once you resolve the small issues in the code syntax. Once the code is working and your data is loaded, try to apply what you have learned about calling data rows and columns. You can always check the console for output. What you see on the screen will show you what you are expected to find in this practice. Once you have understood how to write and execute the code, you will find a link to a Jupyter notebook. You can use this notebook as a starting point for writing a more complete notebook on loading and exploring different types of datasets. So when you see this kind of an image that says GitHub Colab Interactive Notebook, you can just click on that image to get access to this notebook. Once you have completed this part of the assignment, you can save it on your Google Drive and use it later when you need to load a dataset. Now let's try a new dataset. This dataset offers a view of gene expression data. It has samples called by subtype of breast cancer and rows corresponding to genes. Here they're just named gene 1, 2, 3, etc. Once you can technically load and view the data, let's understand what this dataset actually represents. So a typical gene expression table, as you can see here, is the result of an RNA-seq processing pipeline. What this pipeline does is typically take unstructured data, which was unorganized and collection of random reads in a text file, also called FASTQ or FASTA, was mapped onto the genome to produce accurate numbers that represent the quantity of each type of gene. So what you see are sample names, and the rows represent genes. And here you can see the expression level, which is a number that could be normalized for different types of things that we will discuss in a different lesson. We have two specific courses that can help you understand in detail the full process. One of them is called transcriptomics, and the other one is a project called Modeling Cancer Precision Medicine. In that project, you will see the exact definition of the data set that we're going to be using in this practice.